So last but not least is our next speaker, which is Maurizio Porfiri. He's an institute professor. How was that? Is that okay pronunciation? At New York University Tandem School of Engineering. His work's in the general area of complex systems with applications ranging from collective behavior fish to water impact problems. You may not know this, Maurizio has published over 350 journal papers. And another fact about him, he's received the ASME Young Investigator Awards, plural, from 3 different divisions of ASME, which are the control, the vibrations, and the smart structures. So basically, Maurizio broke the Young Investigator Award at ASME. He's also an associate editor of Flow, and we're very happy to have him.
Oh, thank you very much for having me. It's a great pleasure being here and talking about our work, which indeed was published in Flow. And you'll see here this beautiful picture that made the cover indeed. I'll show you a better version at the end. But what I would like to talk to you today about is modeling collective behavior of zebrafish. So zebrafish, for those of you who are not familiar, are pretty cool animals. Many people have called them the third millennium mouse, and they are being used in a lot of studies in which people are performing preclinical research with a hypothesis about different aspects of our well-being. So, for example, they have been used in toxicology, in neurobiology, oncology, tons of studies, and these studies are based on homologies that zebrafish have at the genetic and at the neural level with humans. So tons of studies that have done over the years, 10,000 per year or so. So why are they so popular? There are a number of reasons. From my point of view, I like them a lot because they are very, very social. So you do experiments and they will develop instances of social behavior that are very easy to recognize and to manipulate somehow. Then you can stock very many of them in the lab, which is great. They are less sentient animals than a mammal, and they reproduce very fast, and the time between generations is short. So if you want to run a developmental study to look at the impact of a compound on the development of a subject, you don't have to wait a year, but you'll get the result quite soon. And then, this is a little bit above my pay grade, but the embryos are transparent and the skull remains transparent for a while. So people have done beautiful experiments in which they are able to understand which parts of the brain contributes to an observed behavior. So you can see neuron firing and the animal doing something, which is remarkable. So they have been critical for understanding many, many aspects of behavior. For example, you use them routinely to study fear and anxiety. They have been used to study executive function like learning, memory, and cognition. And then, this is perhaps the most common use, you can utilize these animals for learning about the effect of psychoactive compounds, which is very simple. So you do an experiment and then you would release the drug in the water. The drug is absorbed by the animal, you can control the concentration, and then you score the behavior of the animal. So people have come up with what they call high throughput experiments, in which you record the animal from different angles, you look at the behavior, and then from the behavior, you can infer the emotional state of the subject, and then learn how a particular drug is inhibiting or promoting certain type of behavior within the edogram of the animal. Great. All right. So we do experiments with live animal, but a good chunk of my interest is in developing mathematical models. So these mathematical models are quite neat and helpful. So not only they can help understand the fundamentals of behavior, but also they have an ethical consideration. Because if you develop predictive mathematical models, then you can contribute to the welfare of animals themselves. So although they are less sentient than a mouse, they still are sentient animals. So from our point of view, it becomes really important to devise ways in which we can minimize the number of experiments that you do on a live subject. How do you do that? Well, ideally, some of the experiments, you can run them on a computer. They may not be the most reliable, but they can help you getting pilot data that can serve for performing initial statistical tests. They can help quantify better information about the behavior, so filling data points that were missing in the experimental data set, and then in general be able to reduce the number of animals that you can utilize because you have a better control over the experiment itself. So these kind of in silico experiments, they are relatively unprecedented in the context of behavior. You see them in many different aspects of medical research, but not as much in the study of behavior. Very good. So how do we do this in silico experiment? Well, the simplest thing we can do is start to observe what an animal does. So you can put an animal, one of these guys, in a box, and then you can see how they swim. So this one is a 2D image, so you just see the animal from top. And what you observe is that the animal is displaying this kind of erratic activity in which there are small oscillations in their turn rate, which are accompanied by large turns that correspond to a U or a C turn. Interestingly, you can modulate the onset of the C and the U turn with pharmacological manipulation or simply reducing the level of light by making the animal more comfortable and reducing the stress level. In principle, if you look at a very simple scalar model for the behavior of the animal, you can capture quite a lot with one single variable, which is the turn rate of the animal as it swims. So if you look at the turn rate has this behavior where there are these tiny oscillations, and then sometimes there is a big jump, which is associated with a sudden maneuver, like a C or a U-turn. So if you look at the time trace a little closer, what you observe is that the time trace of the turn rate is centered around 0, which makes sense. The animal doesn't have a compass, so it's just exploring the environment. And you observe that there is a range of oscillation, which is about 0. And on top of it, there are wider oscillations that exceeds 3 standard deviations. So these oscillations are very interesting for those of you that may be familiar with Levy jumps. They are quite similar. Those of you familiar with what happens in financial engineering, they look like jump processes. So what we tried to do many years ago is come up with a stochastic differential equation that could describe the onset of this kind of behavior. So the model that we formulated, we call it jump persistent turning Walker. And it's a simple stochastic differential equation written in terms of the turn rate, in which you have a relaxation that is quantified by the parameter eta, which in the absence of any noise will make the animal swim straight. But in the presence of noise, it will make the animal relax toward 0, but under the presence of this active noise. Noise is critical and is composed of 2 constituents. One of it is the regular, say, white noise that capture the small oscillation about 0, and the other are these wild oscillation, which we capture through a jump process. So if you look at the noise, we have one contribution, which is white noise, this dW, and then the other, which is the product between a Gaussian random variable and then the variation of a Poisson process. So we have sometimes a kick that makes the animal turn, and the intensity of the kick goes with this Gaussian random variable. So in total, you have 4 parameters to estimate. One is the relaxation rate, the other is the strength of the white noise, jump frequency, and jump intensity. So the model is very simple, and we calibrate it on real data of live animals. And you are able to get quite a nice match. So that's just one example, but I believe it's neat that it shows you that we are able to reconstruct not only the central region of the distribution, which is when the oscillations are small, but also the tails of the distribution that corresponds to the C and to the U-turn. So when you look at this QQ plot, the perfection is a nice 45-degree line, and we are pretty close. We can do really well by calibrating these 4 parameters. Okay. Now, we are here to talk about collective behavior. So what I want to do next, which is what we did in the flow paper, is taking this individual model and extend it to the study of a group of animals. So we included 2 different sources of interaction, one of them that have been studied extensively in the literature, which is the set of interactions pertaining to vision. There are many, many papers in the area. I just pick one, but you can have even a paper from the last Nobel laureate Parisi. There are many of them. People looking at mathematical models to capture interaction based on vision. So what you do, you set basic rules, for example, alignment, repulsion, attraction, and then you make the animal interact as a function of how they see one the other. The other part, which is much, much less studied, are hydrodynamic interaction. In the context of hydrodynamic interaction, the paradigm that we utilized is the paradigm developed by Newton and Canso in the Proceedings of the Royal Society in 2012, and it's very, very neat. I like it a lot. So what you do, you associate with each fish a vortex dipole, and then you study the whole interaction problem in the context of potential flow. So what we did in our paper, we combined these 2 aspects. We combined the visual cues with hydrodynamic cues. Now, given the audience, I believe that everybody will be quite happy with the vision model, but looking at the dipole, people may question, is it good or not? Fish are very complicated. Are we really sure that a simple dipole is going to capture the flow physics around an animal? And the answer is yes. I'm going to show you why. So what we did quite recently, we collected experimental data on zebrafish swimming in a water channel. We have very, very many animals. What we can do is track the shape of the animal. We will generate I would call them, let's call it a master shape that is oscillating time, which you can see on the left, to capture the steady swimming of a single animal. And then what we did, we performed a quite simple simulation of a laminar flow by imposing, so it's not a fluid structure interaction problem, it's just imposing the motion of the fish with like a moving wall. What you observe is very interesting, which is that if you consider the mean flow and you take out the background flow, then you are left with the plot in the bottom. So what we see is, surprisingly, a very close representation of the dipole. So if you take the mean, so forget about the oscillations, and take out the background flow, the fish really behaves as a vortex dipole, which is quite nice. So we can use the vortex dipole as a starting point to look at the hydrodynamic interactions between animals. So in practice, what we do is we augment the jump persistent turning walker by changing the relaxation. So rather than having the animal relax toward a 0 angular speed, so that it will go straight, the animal will relax to a non-zero value, which is driven by the combination of hydrodynamic and visual interaction. So that's the idea of the model. So very simple. So we are just adding this omega star and capital omega. One of it defines how the animal interact visually, and the other defines how the animals interact hydrodynamically. Once you solve the stochastic differential equation, you compute omega. And then what we do is we update the position of the animal by utilizing a self-propulsion at a constant speed, and then we impose the advection due to the background flow and interaction with the other animal. So the model is very, very simple in principle. Okay, so if we look at the details of the model, they are not bad at all. So what you do with the vortex dipole paradigm, you associate each fish with a pair of vortices which are placed with respect to the span of the animal. So one vortex is here and the other vortex is below. The strength of the vortices is given by their separation and the self-propulsion speed. So the model is very simple in this regard. And then what you do, you can compute the velocity induced by each vortex dipole. So the notation is a little clumsy, but it works. So we look at two fish, and the little hat on top of it is referring to the index, which is not F. So for example, we study fish 1, so F is equal to 1, the hat refers to fish number 2. So we look at, we have the relative angle with respect of one animal with respect to the other, and then the relative orientation. So you have one piece that quantifies the hydrodynamic interaction. The model is purely kinematic. So we are not looking at added mass. We are simply computing the overall torque by evaluating the gradient of the velocity with respect to the cross-stream direction. And then you have the visual interaction, which is instead composed of one term that defines the alignment, and the other is the attraction. And then we weight the two terms by something that accounts for the impossibility of an animal to see backward, so it captures the cone of vision of the subject. So this is how the model works, very simple. So what we do is we calibrated all the model parameters by looking at experimental data. So we are able to get the 8 parameters that represent a pair of fish, in principle identical, interacting in the environment. And then what we can do next is you can simulate different conditions. So for example, that will be a prospective simulation in which the two fish are interacting under the presence of both visual and hydrodynamic interaction. So what you can see is that they tend, so you can see the interaction is quite complex. It has several phenomena, several parts that are contributing to it, but you see a tendency of one fish to follow the other. There is a tendency of pursuit versus a tendency of swimming side by side, which is interesting. So mathematically, we can score different indicators of social behavior. So one of them is the polarization that quantifies the extent to which the two animals are swimming in the same direction. And that varies between 0 and 1. 1 is perfect alignment between the subjects. So what we observe is that by running the model, we are able to obtain values of the polarization which are comparable to experimental value, and they will be statistically different by chance. The way we compute chance is by shuffling experimental data as if we were considering two fish from different trials so that they don't interact. So that's good. Then we also observe a tendency of the fish to swim in tandem. So there is a pursuit index which is different than chance, so that the two fish tend to swim with one in front and the other in the back. And then since they are identical, they will tend to alternate their configuration. So one is leading, one is following, and they will switch. Then there is a tendency to stay close, which is called shoaling, and that kind of behavior is captured by our attraction. And then this is very, very interesting. We also find that if you look at the speed of the center of mass, the speed of the center of mass is higher than chance, which means that the speed of two fish that are interacting, the speed of the center of mass of two fish that are interacting is higher than the speed of the center of mass of two fish that are not interacting, which is nice because that one is an effect of the hydrodynamic interaction that creates an increase in the speed of the animals. What we did further is we studied the stability of inline swimming, and we were able to do it analytically. So I was very happy to do so. So the way we treated the problem analytically, we introduced a perturbation with respect to the inline configuration, and we were able to express the linear stability matrix in a block diagonal form, in which we isolated a contribution, which is right here, pertaining to the orientation of the pair as a whole, which bears no relevance on tandem swimming, because it's not important if the two animals are swimming with this angle or this other angle. And then we looked at this other block in the bottom that is instead capturing perturbation with respect to delta theta and delta omega. So analytically, we could compute the eigenvalues of this matrix in the bottom and study the conditions that would guarantee stability of inline swimming. What we were able to discover is that there is a wide range of parameters that guarantee stability, provided that the animals are swimming sufficiently far. So the role of the hydrodynamic interaction is basically to repel the animal one from each other and set a minimum distance above which they can afford to swim in tandem. On top of it, when they swim in tandem, you also gain an increase in the speed due to hydrodynamic interaction, and this we were able to observe experimentally. So with this, I conclude the, so I hope it was, yes, so I think that conclude the technical part of the talk. So I would like to thank my co-authors, there was Sean, Peng, and Mert, who helped quite a bit with the formulation of the theory and this recent simulation trying to validate the accuracy of this dipole model. Couple of other words about flow. You can see, I believe, out of the sample of papers we discussed so far, the breadth of the topic. The breadth of the topic is remarkable. So we have applications spanning optics, COVID, and now fish. I believe that that's a unique advantage of the journal, and I iterate the comment of Moran that even in my case, I don't have new insight in fluid dynamics, but I leverage techniques from fluid dynamics and dynamical systems to answer questions about collective behavior. But they are done at a level that it would maybe be premature to present for a biology audience, because you may miss the technical details and the breadth of the conversation that is needed when talking to our community in fluid mechanics. So I'm really looking forward to see more and more papers in flow. It has been a wonderful experience, and yeah, that's about it. So thank you. I hope I didn't drag it too much.
Any questions for Maurizio, either from the audience or from the panel? If we're waiting for one, I might ask a quick question. So is there some way to determine whether the lead fish has information and reacts to the trailing fish? I know the fluid mechanics is like elliptic and coupled, but can you say, yes, there the lead fish now is sped up because he can sense the trailing fish?
Okay, so this is a wonderful question, and it's actually something that we are trying to address in an experimental setting. So that goes back to a paper we published in JFM, so that precedes the creation of flow, in which we try to apply information theoretic techniques to study causal inference in tandem flapping foils. So in principle, we are able to pick up which is the animal that is leading by basically doing the following. What we would do is we would quantify the information that is encoded in the behavior of an animal that helps predict the future of the behavior of the other animal. So we can do that with a nonlinear construct that is called transfer entropy, and by advocating some sort of stationarity of the stochastic processes, we can compute the directionality of the information transfer to some extent. And we were able to show it with tandem airfoils and even looking at the speed of the interaction. So in that example, I had two flapping airfoils one in front and one in the back. And we were making something like the first actively controlled and the other passive. We would observe that from the time traces, I am able to tell that this guy on the back is passive. And I can quantify that the information transfer is at the scale of the advection. If instead I do the opposite, which is I have the one in the back flapping and the one in the front passive, I will also pick up that there is a directional interaction with the one in the back driving the one in front, but the delay at which this interaction happens is on the order of the speed of sound, so much faster. So we are able to tease them out. We are in the process of applying it to fish. Work is ongoing, hopefully get done soon.